So, welcome back to the 5th class of the 6th week, which is the last class of our 6th week. So, today we will be concluding this week with few other biosensors which have been developed for botulinum toxin. So, in the previous class we talked about one of the electroactive sensors. So, overall we have classified this part into 3 aspects. We have talked about the electrochemical sensor. Today, we will talk about the SPR based sensors and immunoassays for multi-analyte detection. So, this is our week 6 class number 5 for week 6, and title of the class will be an optical biosensor assay for rapid dual detection of botulinum toxin A and E. Okay. So, just a recap. So, in the previous class we talked about how this botulinum toxin cleaves a peptide which is developed from the SNAP25 protein, and those of you who needs to recollect. So, this is that SNAP protein where neurotoxin goes and bind, and this is that SNAP protein where this neurotoxin goes and cleave this SNAP protein. So, a peptide is being developed based on that, and this assay electrical signal goes down, and this is how you figure out that botulinum toxin is functional. Today, I will give you a small recap of the SPR technique, if you remember, because this is what we are going to talk about. So, any sort of interaction which is happening on the surface is being captured by the SPR technique. So, if you guys remember, at a certain incident angle or resonance angle, the plasmons are set to resonate with light, resulting in absorption of light at that angle. This creates a dark line in the reflected beam shown in the figure. This is the dark line. The excitation of surface plasmon results in dark line in the reflected beam, and the angular position of the dark line shift as the molecule binding takes place. So, any kind of binding assays will lead to a shift in the dark line. So, there is a, you could see there is a shift like this. The shift can be in both direction, by the way, and this is that angular shift. Based on that, the molecular events are being quantified. Now, today you will observe how this particular technology is being used for botulinum toxin detection. So, this is just a recap for you guys. Now, a small recap here which will help you to understand. So, here you have the botulinum toxin and here have the SNAP protein. So, what is happening is that SNAP25 is, there is an interaction with botulinum toxin A, E, whatever. And this kind of molecular interaction leads to generation of new fragments. As you could see, these are the new fragments which are being generated. Now, this molecular interaction or binding interaction binding is captured via SPR signal. Okay. Now, let us look at it. What is the strategy which is being followed? So, this is your sequence of the C terminal of the SNAP25. This is that SNAP25, slide back. So, this is that SNAP25 protein, and but we are talking about the C terminal. This is that whole C terminal, and these arrows where you see where the different isoforms of botulinum toxins are cleaving it. So, this is where it cleaves it, this is where it cleaves it, this is where botulinum C cleaves it, this is where botulinum A cleaves it, this is where botulinum E cleaves it. Okay. Now, if you pick up this segment out here, this segment. So, they developed an antibody against it. So, for botulinum E, this is the antibody. Similarly, if you pick up this segment, which is all the way to Q, right. So, you have monoclonal antibody MABs for botulinum toxin A. Okay. So, here you have botulinum toxin A and E, and C is out here. Okay. They have not shown any antibody produced against it. Arrowheads, as I have already mentioned. The new C-terminus domain of botulinum E and botulinum A cleaves are illustrated, and the peptide sequence are used to generate monoclonal antibodies 11C3. This is against E, and this one is against A. Okay. So, now you have the antibodies at your disposal, and these antibodies will bind to the toxin fragment. So, that means, now going back to the logic what I was trying to tell you, now replace this interaction with, say for example, monoclonal antibody MABs and neurotoxin, or any kind of, in this case we are talking about botulinum toxin, depending on which version of monoclonal antibody you are working, A, E, C likewise. Okay. So, this interaction is being captured on a SPR chip, and these are, as I mentioned in SPR, these are label free. There is no labeling, because you are only evaluating or quantifying the interaction. Okay. So, now coming back, so you have the monoclonal antibody, see this is 11C3A, which is against E, and this is the peptide interaction. Okay. Now, as I was mentioning, botulinum toxin E, and here you have the monoclonal antibody against it. Okay. MAB 11C3, MAB 11C3. Now, what we are going to see here is cleavage, different contact point. So, this is cleaving, this is coated with SNAP. So, if this is all coated with SNAP and you are allowing this antibody to run through it, what it will do? It will cut it at specific points, and those points are out here, this 180th residue, where this particular peptide is going to, or this particular toxin is going to cut it. Okay. Scheme illustrating the general principle of the assay. In the first step, botulinum toxin E sample is injected over SNAP 25 sensor chip for a predetermined contact time. This is important. In the second step, the neoepitope produced by botulinum toxin E activity is detected using MAB 11C3. So, what is happening here is that this is cutting them into fragment, and there are fragments for which antibodies have been raised. That antibody is this antibody. So, this antibody is now going to bind to that fragment, and this binding assay between MAB 11C3 and SNAP fragment is what you will study in the SPR cycle. So, first of all, this is A, this is B, this is C, and this is the E. This is assay specificity, where you are specifically assaying it, the binding signal of MABC3A to all the flow cells, but this particular antibody will only bind to the fragment which are generated by this particular isoform of botulinum toxin, and these are different isoforms. Every different isoform cut it into different spots. So, if you look at this picture, so every isoform is cutting it at different point. So, unless you have another antibody which is for botulinum toxin A, which is MAB10F12, this won't work. This will only work for MAB11C3A will only bind to the fragment of SNAP25, which has been cut by botulinum toxin E. Okay. So, that is why here you see the maximum signal. Okay. Now, what they are doing is, so this is no toxin, a baseline you see, there is absolutely no toxin. Now, you are adding the toxin out here and you start to see a molecular interaction. So, there is a signal out here, fine. Then you use A, you do not see a signal here, but you see a signal later. So, it means this has also the other one co-detection. So, we are talking about you detecting both of them. So, it means you need to have the other antibody, which is 11C3A is for E, whereas yes, and whereas the other one, which is MAB10F12, yes, this one is for A. Okay. This one is for A. Okay. So, what you are trying to do here, you have a kind of a sandwich situation, where you can detect both botulinum toxin A and botulinum toxin E, provided you have on the flow cell, you have the antibodies for both of them. So, what will happen in global picture? So, we know that SNAP protein in the synaptic vesicle is being cut at different point by different isoform of botulinum toxin, and botulinum toxin has isoform like A, E, C, B, so on and so forth. Okay. Now, A will cut it at a specific spot and will generate a peptide. That peptide will be used to develop an antibody. In this case, for E, as we have seen, the antibody monoclonal antibody generated is MAB11C3. Fine. Similarly, if you cut the SNAP with botulinum toxin A, the peptide which will be generated will be used for the production of another antibody. In this case, see MAB10F12, which is can interact with botulinum toxin A. Okay. Now, what you are trying to do is, you are doing a sandwich assay where you want to detect both. Say for example, your sample has A plus E. So, automatically you need the antibody against A, which is 10F12, and you need the antibody against E, which is 11C3. Both these antibodies you will be needing for your assay. So, this is the first signal you are getting is for E, the second signal you are getting is for A, and the summary signal you are getting for A as well as E. This is what is you are getting in the SPR chip. Now, if you look at the signal of SPR while I was trying to, so this is what you see, the shift. So, you see two shifts. So, you see second one coming after. So, this one is for say E and then this one is for A, depending on which botulinum toxin we are talking about. And if you follow this picture, now I will take you to the real life picture. This is like this, right. So, this is for E, this is for A. So, at one point of time, without any label, you could detect different fragments of SNAP25 using specific monoclonal antibodies against those cleaved peptides. So, that means indirectly you are detecting the botulinum toxin A and E in one single SPR chip, because there are two molecular binding events which are taking place, because the peptide fragment which is being released by the cleavage or by botulinum E binds to the first antibody, which is MAB11C3. Similarly, the cleavage of the SNAP25 done by botulinum toxin A binds to MAB10F12. So, in a way, the whole assay is between the binding of the peptide cleaved off from the SNAP25 and the monoclonal antibody raised against it. So, the only job of botulinum toxin here is to cleave the SNAP peptide. Now, if you once again revisit this one. So, what is happening here is, in the first step, botulinum E sample is injected over SNAP25 sensor chip for predetermined contact time. In the second step, the new epitope produced by botulinum toxin E activity is detected using monoclonal antibody 11C3. So, imagine if this chip also has at another point the other antibody, which is this antibody MAB10F12 plus MAB10F12, then you will be able to detect that one, provided you are running A, because A is specific to this one, whereas E is specific to this one, right. So, these are the state of the art currently in botulinum toxin detection. And these, as I has mentioned while we talked about SPR chip, SPR is one of the most popular method currently being employed across the world for botulinum toxin detection. From here, we move on to a second level where we will talk about multimodal detection. So, for the food, which is one of the challenging part, biosensor detection of botulinum toxoid A and Staphylococcus enterotoxin, which sometime in short they call it ACB, S here, E here and B here in food. So, if you look at the immunoassays were developed for simultaneous detection of Staphylococcal enterotoxin B, which is ACB, and botulinum toxoid A in buffer with limits of detection of 0.1 nanogram per ml to 20 nanogram per ml respectively. So, 0.1 nanogram for ACB and 20 nanogram per ml is for botulinum toxin A. Okay. The toxins were also spiked and measured in a variety of food samples, including canned tomato, sweet corn, green bean, mushrooms and tuna fishes. The toxin Staphylococcus enterotoxin SCB and botulinum toxin A are responsible for food poisoning, as we have already studied about it, and have potential to be used as biological warfare agent. With the current toxic dose for aerosol form is 0.02 microgram per kilogram body weight and 0.07 microgram per kilogram respectively for different kind of each one of these ACBs and the botulinum toxoid. There is a need for rapid method for monitoring food, water and air sample for both natural and intentional contamination by these toxins. Now, immunoassay chip, what we will be discussing now, will take you to one of the sandwich chip, if you guys remember. I will just take you back to that, just to give you an idea. Just a small recap. So, we talked about these kind of immunoassays which are being done. So, now let us visit what is the current state of the art of these assays. So, biotin-ligand capture antibodies in phosphate buffer saline containing twin, these are all different kind of detergent, were patterned on neuroavidin functionalized slides using PDMS flow cells. So, PDMS is a common element for developing microfluidic chambers and biomems chambers. Okay. Each slide was patterned with 2 columns specific for chicken immunoglobulin Y positive control and 5 specific for botulinum toxin A and 5 for ACBs. 20-minute sandwich immunoassay, as I was mentioning, sandwich immunoassay were performed. Fluorescently labeled tracer antibodies Psi5 chicken immunoglobulin Y, and 10 microgram per ml each of Alexa-Flor 647 anti-SCB and Psi5 anti-botulinum toxin A in PBS containing 1 milligram of bovine serum albumin is used to detect the bound targets. Now, this is how the result looks like. So, these are the flow cells where you have rabbit anti-botulinum toxin, RB anti-ACBs. Okay. Now, charge coupled devices image of simultaneously 20-minute assay for the sample containing botulinum toxin of different concentrations 0 to 500 nanogram, as you could see. As you see, so here you have the highest one and the signal is going down. So, pretty much you see a faintest signal somewhere around 10 nanogram. Okay. 0 to 500 nanogram and ACB 0 to 7.5. Okay. So, you see the maximum signal at 7.5, and if you go somewhere around out here, if you follow this line, you do see certain signal, but after this signals are fading. So, up to this you kind of see some of the signals. Okay. Now, sample channel numbers are indicated by the numbers in the bracket. RB stands for the rabbit, botulinum toxin, IgG immunoglobulin, and all the other short forms are given there. So, this is how the modern day assay systems look like. So, these are all micro pattern on a PDMS polydimethoxysilane chip, and at these different spots of this flow cell you have immobilized the antibodies or your toxins or your substrate, and these are all done by microprinting or micromachining techniques or micro spraying at specific locations. Okay. And this is what you get further based on the concentration, normalized intensity. So, these intensities are being normalized, and this is how you get the data from this kind of situation. And further, if you look at the limits of detection out here, this is the kind of at different pH. Of course, this is very important that you follow the pH. This is the kind of detection you get in different food samples, say 20 nanogram, 50 nanogram, 20. So, depending on tomato juice, whole tomatoes, so mushrooms, sweet corn juice, green bean juice, and tuna fish. Tuna fish mostly comes in the cans. And there is always a possibility of some of these contamination existing in them. So, this is the kind of numbers we are looking at. In the last class, I told you that it all depends at what is your detection limit, and detection limit should be unambiguous. You cannot, because when you are at the commercial level, if this is, if I say 0.1, then it should be almost 99%, it should be 0.1, maybe more than 99%, it should be 0.1. So, this is what we talk about as multi-analyte detection. Assays are simple to perform, show no cross reactivity, are rapid, simple, require little or no sample pre-treatment or pre-concentration. The main advantage of the array biosensor over existing technology is its ability to detect multiple analyte in multiple sample simultaneously. So, time, specificity, these are of extreme importance. Multi-analyte sensors increasingly appear in the literature, but most have yet to look at matrices more complex than buffer. The ability to carry out multi-analyte detection in the complex sample is a clear advantage for screening food, water or air samples for a hazard either naturally occurring or deliberately introduced. So, as I am mentioning that most of the biosensor development process across the world are now going for multiple techniques and multiple analyte at the same time. It is a lot of things eventually will be AI driven. They will be transformed as AI driven systems, where I will have to make a call in a sample. There will be multiple contaminants, and you should have multiple modalities to detect those contaminants, and you should be able to make a call that how to handle the sample. So, multi-analyte detection and multimodal detection technology, as long as we could couple both of them synergistically, we will get tons of data, but of course, there you will be needing a lot of computational power, will be needing the help of artificial intelligence algorithms to develop and detect and to make a call what will be our decision making under such situation. So, with this lecture, I close the 6th week, and next we will move on to other different form of analyte. So far we have talked about botulinum toxin, we have talked about other different kind of toxins which are present. So, slowly we will move more towards all the detection systems. So, I have already given you a very clear cut understanding what are the different areas, what you need to understand before you develop as a professional biosensors person who can develop or design biosensors. Thank you.